Welcome, future well-informed individual, to another deep dive. Have you ever found yourself wondering, really wondering, how much we can truly trust the images that shape critical medical decisions? Think about an MRI scan guiding surgery, or maybe a PET scan tracking cancer treatment. We tend to rely on these high-tech images, sort of assuming they're giving us perfectly consistent, accurate information. But it's definitely not magic. It's meticulous science, and sometimes, well, that science faces some surprisingly tricky statistical challenges.
Exactly. We often take the results of medical imaging almost for granted, but behind every quantitative imaging biomarker or QIB, an objectively measured characteristic from an image, there's this really rigorous process.
Right.
It's not just about getting a pretty picture, it's about making sure those measurements, the numbers, are reliable enough to actually guide patient care or track disease or even help approve new drugs.
Okay, so today we're diving into a fascinating paper by Huang et al. From 2014. It's essentially a blueprint, you could say, a master guide for how we approach a meta-analysis on these complex imaging procedures. Our mission for you, our listener, is to decode why this statistical deep dive is so critical, look at the clever ways researchers tackle its unique problems, and figure out what it ultimately means for the future of medical diagnosis and care. Okay, let's pull back the curtain on this vital science. So let's start with this term. Quantitative Imaging Biomarker, QIB. Sounds incredibly important. Can you break down what it actually is and where it fits into the grand scheme of patient care?
Sure. So QIBs are basically measurable features we extract from medical images. Think of them as the precise data points we get from a picture, things like the exact size of a tumor, maybe how much blood is flowing to a specific area, or the metabolic activity within a lesion.
Okay, so they give us numbers, not just descriptions.
Exactly. They give us numbers. And these numbers play many roles. They can help stage a disease, guide treatment decisions, and even serve as objective endpoints in clinical trials for new therapies. But here's the crucial part. Before any QIB can be widely accepted, the imaging procedure that produces it absolutely must demonstrate acceptable technical performance.
Technical performance, that sounds like the bedrock everything else is built on. What does that specifically entail? What are we measuring there?
Well, it means ensuring accuracy, reliability, and critically repeatability and reproducibility.
Okay, repeatability and reproducibility.
Yeah,
The difference. Good question. Repeatability asks, if we scan the same patient under identical conditions twice, do we get essentially the same result? It's about consistency within a single setup, like using the same scanner, same operator, same day.
Got it. Same setup consistency.
Right. Reproducibility then goes a step further. It asks, do we get consistent results even if we change some factors, like the specific piece of equipment, maybe a different operator, or even imaging at a different hospital site?
Ah, okay. So repeatability is tight control. Reproducibility is more real-world variation.
Precisely. And if these performance metrics aren't sound, if the measurements are too noisy or inconsistent, then the QIB's clinical utility is really compromised.
Meaning?
Well, it could mean a patient isn't diagnosed correctly, or maybe a drug that is actually working might be prematurely abandoned because the imaging results are just too variable to show the effect clearly. It undermines the whole point.
And this is where the paper highlights a really big challenge, isn't it?
Right.
These individual studies that try to evaluate this technical performance, they often have surprisingly tiny sample sizes, sometimes as few as, 10 to 20 patients?
Yes.
That just feels like, a too small a number to make broad, confident conclusions from.
You've hit on the central problem. It's like trying to judge the performance of an entire orchestra based on hearing just one or two musicians play. It raises this crucial question. How do we overcome the limitations of these small individual studies? How do we get a much more robust, trustworthy understanding of an imaging procedure's true performance?
This is precisely where meta-analysis shines. Instead of just a qualitative narrative review, which can be subjective, someone just summarizing a few articles they like, may be prone to bias. Meta-analysis takes a systematic review a powerful step further.
How so?
It quantitatively combines the results from multiple independent studies. It pools the data together statistically to provide a single summary estimate of these performance metrics, like repeatability or reproducibility. It truly is the strongest methodology we have for synthesizing evidence from diverse studies, basically turning many small signals into one loud, clear message.
Okay, that makes sense. Meta-analysis as the synthesizer. So if it's our gold standard, how do you even begin to gather the right ingredients, the studies themselves, for this powerful statistical recipe? It sounds like it must be more involved than just a quick search on PubMed or Google Scholar.
Oh, absolutely. What's fascinating here is the sheer rigor required for the systematic review process before you even get to the meta-analysis number crunching. First, you have to formulate a research question with, well, surgical precision.
Precision like how?
It means specifying 3 key things very clearly. 1, the clinical context. Are we, say, screening apparently healthy individuals, or are we monitoring cancer treatment response? Very different scenarios. 2, the class of imaging procedures. Are we looking at FDG-PET scans, a specific manufacturer's CT scanner, certain image acquisition settings? You need to define the technology.
Okay, context and technology.
And 3, the specific performance metric you're interested in. Is it the repeatability coefficient, the RC, or something else? For instance, if you're using FDG-PET to assess treatment response in cancer, a well-defined RC could establish a vital threshold.
A threshold.
Yeah, a cutoff point. Any change in the measurement above this number might indicate a true treatment response, whereas changes below it could just be random scanner noise or biological fluctuation. Without that clarity in the question, you're building your whole analysis on shaky ground.
I can imagine there are some tricky real-world factors that make getting this data a challenge, though. The paper mentions things like asking patients to undergo repeat scans, radiation exposure, or even traveling to different hospitals.
Absolutely. We have to be realistic about what's practically achievable and ethically appropriate. Conducting true repeatability studies might involve patients getting repeated radiation doses, which you want to minimize, or maybe they need washout periods for injected contrast agents, which can be burdensome.
Right.
And true reproducibility might require subjects to travel between different imaging sites, which is rarely feasible for every single study participant. These real-world constraints mean researchers often have to work with data that isn't perfectly ideal.
So the data might be a bit messy sometimes.
It can be, and sometimes that observed variability, that messiness itself, can be quite informative about real-world performance. There's also this delicate trade-off when formulating the question. If it's too narrowly focused, you might find too few studies to even do a meta-analysis. But if the question is too broad, you might lump together studies that are too different, introducing too much heterogeneity, we call it, which makes interpreting the combined result really difficult. It's a balancing act. And the paper wisely suggests that if the meta-analysis is ultimately for regulatory approval, like for a new medical device, consulting early with the FDA is highly recommended to make sure everyone's on the same page.
So once that meticulous question is finally set, then comes the real detective work. Finding the actual studies. And you mentioned earlier, it's not just about sifting through the published scientific papers we usually hear about.
That's right. The search has to be comprehensive. You can't limit it just to published literature. Why? Because of something called publication bias.
Publication bias.
Yeah, it's this tendency for journals to preferentially publish studies that show statistically significant or, positive or favorable results. Studies with less exciting findings or null results where maybe they didn't find a difference might never get published, even if the science was perfectly sound.
So they just sit in a file drawer somewhere.
Effectively, yes. Or they only get presented briefly at a conference. So, to combat this bias, researchers conducting a systematic review must dig deeper. They need to look for unpublished information in places like meeting abstracts, clinical trial registries. Clinicaltrials.gov is a big one, technical reports, regulatory summaries from agencies like the FDA, like their 510K summaries for devices.
Wow.
And sometimes it even involves directly contacting investigators who are known to work in the field or exploring these growing imaging data repositories like the Cancer Imaging Archive. The more thorough and exhaustive this search is, the less potential there is for that publication bias to skew the overall picture you get from the meta-analysis.
Okay, so after all that digging, you've got this potentially huge pile of studies, published, unpublished, abstracts, reports. How do you then narrow it down to the ones that are truly relevant and, importantly, high quality enough to include?
This is where clear inclusion and exclusion criteria become your filter. You defined very precisely what makes a study suitable for your specific research question.
Like a checklist.
Exactly. For instance, going back to the FDG-PET example for repeatability, the criteria might specify things like the study must measure 18F FDG uptake. It must be in malignant tumors. They have to use standardized uptake values. SUVs, follow uniform acquisition and reconstruction protocols. And critically, for repeatability, they must have used the same scanner for both the test and the retest scan.
Gotcha. Very specific.
Very specific. The paper uses the Delangen et al. Meta-analysis as a great example. They started with 8 potential studies from their initial search, but after applying their rigorous criteria, they ended up removing 3 of them, leaving just 5 studies that perfectly matched their question.
Okay.
And beyond just relevance, you also need to assess the quality of each study you're considering. You look for potential design flaws, any issues in how they did their statistical analysis, and you have to make sure there's no data overlap, like the same patient data accidentally getting reported in multiple different publications from the same research group. It's all about selecting studies that truly align with your research question and meet high scientific standards.
That's a huge amount of preparatory work just to get the right ingredients.
Yeah.
So, okay, what does this all mean when we finally have our carefully selected studies? Now we need to combine them to get that powerful quantitative summary. But the paper points out this major hurdle we touched on, those small sample sizes.
Indeed, this is where many standard off-the-shelf meta-analysis techniques can really fall short. Most traditional methods, the ones used for decades in other fields, make a fundamental assumption.
Which is?
They assume that the estimates coming from the individual studies, like our repeatability coefficient, the RC, are approximately normally distributed. Think of this as assuming your data points, if you plotted them, would neatly fit into that symmetrical bell curve shape.
The famous bell curve.
Right. But with our typical small study sizes and imaging performance, remember, often just 10 to 20 patients, this assumption is frequently violated. The data often just doesn't follow that nice bell curve, especially for metrics like variance components that make up the RC.
And what happens when that assumption is violated? What's the consequence?
Well, simulation studies mentioned in the paper clearly demonstrate that when studies are small and this normality assumption doesn't hold, the confidence intervals you calculate using those standard methods are often less than their nominal level.
Hold on, less than their nominal level. Can you help us unpack what that means for someone who isn't a statistician? What's the practical impact?
Absolutely. It's a key point. When we set a nominal level for a confidence interval, say 95%, we're essentially making a claim. We're saying we are 95% confident that the true underlying value of this metric lies within this calculated range.
Yeah.
That 95% is our target for reliability.
Okay, our desired confidence.
Exactly. But if the actual coverage probability, which is how often that interval really captures the true value in practice, drops significantly below that target, say down to 70% or 80%, because our assumptions were wrong.
Then our 95% confidence wasn't really 95% confident at all.
Precisely. It's like making a weather forecast that claims a 95% chance of rain, but in reality, only gets it right 70% of the time. You think you're getting robust, highly confident information from your meta-analysis, but you're actually getting overconfident predictions. These inaccurate confidence intervals could lead to poor clinical decisions or misguided research directions.
Wow. So this isn't just some arcane statistical issue. It has direct, profound implications for how much we can trust those medical imaging results in practice.
Absolutely. It cuts right to the core of reliability.
Okay, here's where it gets really interesting then. If the standard tools are flawed for these small imaging studies, how do statisticians adapt? How do they ensure the reliability of the meta-analysis despite these fundamental challenges?
This brings us to some truly innovative solutions the paper describes, ways to handle this non-normality. One really powerful approach. If the exact statistical distribution of the performance metric is known and can be mathematically described or analytically tractable, as statisticians say, is to use exact likelihoods instead of relying on that potentially faulty normal approximation.
Exact likelihoods.
Yeah.
So using the true math shape of the data.
That's a great way to put it. Most standard methods try to force your data into that generic bell curve mold. But with exact likelihoods, instead of a generic mold, we use the true custom-made mathematical shape that the data actually follows. For example, it turns out the squared repeatability coefficient, RC squared, actually follows something called a gamma distribution, which has its own specific known mathematical form, quite different from a normal distribution. And simulation studies showed that using these exact likelihoods, using the correct mathematical shape, significantly improved the coverage probabilities of the confidence intervals. It brought them much closer to that desired 95% level, even with those challenging small study sizes. It's like using a custom-made key for a specific lock, ensuring a much better fit and a far more accurate result.
That's brilliant. So don't force the data to fit the model. Make the model
Yeah.
fit the actual data distribution. That makes a lot of sense. What about dealing with differences between studies? Because they won't all be identical, right?
Exactly. No two studies are ever perfectly identical. There's always some variation. For handling this, we often use random effects models in meta-analysis. These models explicitly account for that inherent variability, that heterogeneity between studies. And for these models, especially when normality is questionable, the paper discusses using the expectation maximization algorithm, or EM algorithm.
EM algorithm sounds
No,
complex.
it's an iterative technique, quite powerful. Imagine you're trying to piece together a puzzle, but some pieces are missing or blurry. The EM algorithm is like a clever strategy. It makes an educated guess about the missing information. That's the expectation step. Then, based on that guess, it figures out the most likely overall picture, the maximization step.
Okay.
But it doesn't stop there. It uses that new picture to make an even better guess about the missing pieces, and it repeats this E-step, M-step process over and over again. Each cycle refines the estimates, allowing it to converge on the most likely overall result, even when the underlying data distribution isn't simple or perfectly normal. It's very robust in tricky situations.
And the paper doesn't just talk theory. It actually applies these different techniques to that real-world example you mentioned, the DeLongen meta-analysis of FDG-PT test-retest repeatability. What did they find when they compared these different statistical methods on the same data?
This is where the rubber meets the road, and it's a fantastic illustration. Using the standard fixed effects meta-analysis, the one relying on the normal approximation, the estimated overall repeatability coefficient, the RC, was 0.79.
Okay, 0.79.
But when they applied the fixed effects method, using the exact likelihood, the one that correctly accounts for the small sample size non-normality, using the gamma distribution, the estimate jumped significantly to 1.53.
Wow. From 0.79 to 1.53, that's almost double. That feels like a massive difference.
It is a massive difference. It's not just noticeably different, it's a profound shift in the result.
Can you maybe paint a picture of what that actually means for a patient or a doctor? A jump from 0.79 to 1.53 must have real clinical implications.
Oh, absolutely. Let's think about that cancer patient example again, monitoring tumor response to chemotherapy with PT scans. If doctors were relying on the standard, less accurate statistical method that gave an RC of 0.79, they might interpret that as meaning there's a fair bit of inherent noise or variability in the scanner's measurements. So if they saw, say, a 10% reduction in the tumor's metabolic activity, the QIB value, they might dismiss it. They might think, well, with an RC of DORA 0.79, a 10% change could easily just be random scanner fluctuation, not real shrinkage.
Okay, so they might think the treatment isn't working when it actually is?
Potentially, yes. But now, consider the result from the more precise exact likelihood method, the one that gave an RC of 1.53. This higher RC value implies more inherent variability. Paradoxically, this might seem worse, but it sets a different threshold. Now, that same 10% reduction might fall below the threshold defined by the RC of 1.53, reinforcing the idea it could be noise. However, a larger change, say 20%, might now confidently exceed the 1.53 threshold and be recognized as true treatment response.
So the interpretation of the change depends critically on having an accurate RC value.
Exactly. The point is, the magnitude of change needed to be confident it's real depends heavily on that RC estimate. Using the wrong statistical method gives you the wrong RC, which leads to the wrong interpretation of whether the patient is responding. You might stop an effective therapy too soon or continue an ineffective one for too long. It underscores how absolutely critical it is to use the appropriate statistical method for the specific data you have.
That's a really clear explanation, thank you.
Yeah.
And did the analysis show anything else interesting?
Yes. The random effects methods, including the EM algorithm approach, produced estimates somewhere in between, around 1.25 to 1.34, and importantly, they had wider confidence intervals. This acknowledges that inherent variability between the different studies included in the meta-analysis. The analysis also performed meta-regression, trying to explain why the studies differed. They found interesting links. Factors like having a higher average baseline tumor activity, SUV mean, were associated with a higher RC, more variability, while having more tumors located in the chest area was associated with a lower RC, less variability. This really highlights the power of meta-regression to dig into the sources of heterogeneity when you have detailed information or descriptors available for each study.
So we've talked a lot about combining the summary data reported in different studies, but what if you could actually get your hands on the raw data? The individual patient measurements from all those original studies, would that be even better, a sort of ultimate meta-analysis?
That's a great question. If we connect this to the bigger picture, having access to individual patient-level data, or IPD, offers some distinct advantages, although perhaps not always in the way people first assume. For calculating simple overall summary metrics, like the average RC, using IPD doesn't always lead to major gains in statistical efficiency compared to combining the summary stats properly.
Oh, okay.
Yeah.
So maybe not a huge boost for the main number.
Not always for the main pooled estimate itself, but where IPD really shines is for meta-regression. Remember how we just talked about exploring why results differ between studies?
Yeah, looking at tumor location or baseline activity.
Right. IPD allows you to directly calculate metrics or explore relationships that might not have been reported at all by the original investigators. More importantly, technical performance often varies based on subtle patient-level characteristics. Things like the contrast of a specific tumor against its background, the exact shape or texture of a lesion, the baseline tumor size for that patient, or even physiological factors like how well a patient held their breath during the scan or how much they moved.
Things you'd lose if you only had the average for the whole study group.
Exactly. Using individual patient data for meta-regression gives you substantially greater statistical power to detect those associations between patient-level factors and the imaging performance. You can uncover subtle but important nuances that would be completely obscured if you only relied on study-level summary statistics, like the median tumor volume for an entire study cohort. It's like having a magnifying glass to see the details, not just the overall average. You get to see the forest and examine each individual tree within it.
This deep dive has really highlighted the intricate complexities, the layers involved in ensuring medical imaging is truly reliable. We focused a lot on repeatability and the repeatability coefficient because it had that nice tractable map. But what about other critical aspects of technical performance, like reproducibility across different scanners or sites, or assessing systematic bias in measurements? Do the same challenges and those sophisticated solutions apply across the board?
That's a very important point. The general process for conducting the systematic review, finding the studies, filtering them, assessing quality remains pretty similar for other metrics like reproducibility or bias. You still need that rigorous upfront work. However, the statistical analysis often becomes significantly harder.
Harder how?
Well, for many of these other important metrics, their exact statistical distributions are not known or they're not analytically tractable like the squared RC was with its gamma distribution. This means some of those more advanced exact likelihood approaches, the ones that gave us such a big improvement for RC, simply aren't feasible. You can't use the custom-made key if you don't know the exact shape of the lock.
Ah, okay. So the clever math trick doesn't always work.
Right. This raises a tough question. What do we do when even our clever modifications, our best statistical tools, don't perfectly apply? This is definitely an area that's ripe for future statistical research. There's a need to develop more robust methods, perhaps including non-parametric meta-analysis techniques, which are methods that don't rely on making strong assumptions about the data's underlying distribution at all.
So after this fascinating dive into the statistical trenches, what's the big takeaway for the future? How can we collectively make sure the medical imaging we rely on continues to improve, that it remains trustworthy, and ultimately serves patients better?
The paper offers some clear, actionable recommendations looking forward. First, and perhaps most fundamentally, investigators simply must be encouraged to conduct and, importantly, to publish more high-quality imaging performance studies. And crucially, they need to do so with complete and transparent reporting. Following reporting guidelines, like the PRISMA statement for systematic reviews, it's essential here, ensuring every detail is laid bare for scrutiny and potential inclusion in future meta-analyses.
More data, reported clearly. Makes sense.
Second, there needs to be greater coordination among investigators and professional societies. Groups like the Quantitative Imaging Biomarkers Alliance, or QEA, which incidentally was a driving force behind the research in this very paper, are vital. They work to standardize protocols and promote comparability across studies. If everyone measures things in a similar way, the data becomes much easier and more meaningful to combine later.
The standardization is key.
Absolutely. And finally, these challenges, like the small sample sizes and non-normal data, they actually present incredibly fertile ground for statistical researchers. There's a real need and opportunity to develop entirely new methodologies. This could include things like multivariate approaches, allowing us to analyze multiple different QIBs extracted from a single image simultaneously, understanding how they relate to each other. The bottom line is knowledge is most valuable when it's not just generated, but also understood and applied correctly. And critical thinking about the quality of evidence is just essential in this world overflowing with information. We're constantly striving for higher precision to improve patient outcomes.
What a journey through the statistical underpinnings of medical imaging. We've seen how critical these quantitative imaging biomarkers are, how meta-analysis serves as the gold standard for synthesizing the evidence we have, and really the ingenious ways researchers are tackling these unique challenges, especially the problem of small study sizes in evaluating technical performance. It's a powerful testament, I think, to the fact that even in the highest of high-tech medicine, the basics, good data, meticulous methodology, sound statistics are absolutely paramount.
Indeed. It's through meticulously combining evidence from multiple studies and applying these advanced, appropriate statistical methods that we move closer to a truly reliable understanding of medical imaging performance. The goal is to ensure that when you or a loved one receives an imaging report, you can have confidence that the numbers truly reflect the underlying biology and aren't just statistical noise or an artifact of using the wrong analysis method.
So the next time you encounter a medical image, or maybe hear about a promising new diagnostic tool, perhaps take a moment to consider the profound layers of scientific rigor that lie beneath the surface. It's not just the impressive scanner itself, but often the immense, quiet work of statisticians and researchers carefully determining its reliability that makes that technology truly useful in caring for patients. How much painstaking analysis went into figuring out its accuracy? And maybe consider this. What other fields, perhaps completely outside of medicine, might benefit from this same kind of deep, quantitative scrutiny of their data, using meta-analysis to truly understand what's signal and what's just noise? We leave you with that thought to mull over until our next deep dive.